Good evening, everyone, and thank you for joining us. I'm Laura MacNeill, and I'm the CEO of the CMT Research Foundation. It's a privilege to welcome each of you to tonight's webinar, a gathering of people that are united by hope, urgency, and a belief in scientific breakthroughs for Charcot-Marie-Tooth disease. At CMTRF, we're a patient-led nonprofit that's laser-focused on funding and accelerating the discovery and treatments and cures for CMT. Our mission is personal. We exist to bring real therapies to millions of individuals and families affected by CMT within our lifetime. Even though this disease affects over 3 million people worldwide, for far too many years, solutions have lagged behind the needs. That's why CMTRF takes an entrepreneurial approach, working arm in arm with our scientists and companies and donors to ensure that research leads rapidly from the lab to meaningful treatments. Every dollar we invest is held to milestones, outcomes, and most importantly, the potential to change lives. In 2025, that sense of urgency and partnership has never been more important. Our collaboration with Asha Therapeutics is a model of what's possible when focus and innovation align. The research we're supporting targets CMT2A, which can be a severe form of the disease, and investigates SARM1 inactivation as a transformative therapeutic strategy. This effort demonstrates that when patients, funders, and scientists work together, progress is accelerated for everyone. So tonight, it's my pleasure to introduce Dr. Bradlee Heckmann, the President and Chief Scientific Officer at Asha Therapeutics. Bradlee is an acclaimed neuroimmunologist and drug developer whose career has advanced our basic understanding of neurodegenerative diseases and pioneered new approaches to treating them. He's held key academic posts, contributed foundational research in neuroinflammation, and now through Asha is translating these insights into potential breakthrough treatments for CMT and other related conditions. Dr. Heckmann earned his PhD at the Mayo Clinic College of Medicine, served as an assistant professor at the University of South Florida Neuroscience Institute, and co-founded Asha Therapeutics, leading the charge on innovative therapies grounded in deep molecular expertise. Please join me in welcoming Dr. Bradlee Heckmann, and thank you for being part of the future for CMT research.
Well, Laura, thank you first and foremost for the wonderful introduction. Might be a little overstated, but we're certainly grateful for both you and for the CMT Research Foundation as a whole, and of course to the patient and family communities that are suffering from the over 100 forms of CMT that exist to date. And so we're trying to make a small little impact in how we go about treating Charcot-Marie-Tooth, in particular, as you mentioned, CMT type 2A, but hopefully impactful beyond just the 2A subtype into the all axonal forms of CMT. And so it's a pleasure to be here with everybody tonight. I would love the opportunity to walk you through a little bit of the basic science behind CMT2A, how it differs from more common forms like CMT1, and our methodology for being able to try to therapeutically engage this protein SARM1 that Laura just mentioned as a therapeutic avenue. And let me see if we can get this up. Hopefully we can all see this. There we go. And so we've been heavily focused at Asha Therapeutics on rare diseases and rare neurodegenerative disorders as a whole. Our major underpinning advancement that we have made is the ability to selectively design new medicines for highly therapeutically advantageous targets. And so that's allowed us as a smaller company to be able to move new therapies forward in the developmental process from concept all the way through to a new medicine that we can start testing. And so we've been able to do this a few different times, and that's no different than what we're trying here with SARM1. But before I get to SARM1 and where we are engaging SARM1 for therapeutic benefit, first we need to understand the differences between the varying forms of CMT and, of course, other motor neuropathies like diseases including ALS, for example. where these pathways are also implicated. And so our major focus in the CMT realm is, of course, CMT type 2A, which is this form of axonal degeneration that's characterized by axonal degeneration. And so in your central nervous system and in your peripheral nervous system, we know in CMT that you end up with loss of signal movement. So essentially, you lose the communication between your brain and your peripheral system. So that includes your muscles, and in certain cases, nerves at the skin level, so within your dermis and epidermis, and you end up with neuropathy, obviously motor dysfunction, and a host of other symptoms, numbness being one of those as well. And we know that in a normal setting, you have a healthy neuron that is very nicely organized. It's doing its job, communicating the signal from your brain all the way down to these peripheral tissues. And this neuron is not just a long wire, if you will, but it's actually an insulated wire. And so we have these pieces of lipid that surround this neuron that we call myelin. And so CMT type 1, for example, is a demyelinating form of CMT where you actually lose the integrity of this myelin. And essentially what you do is you break down the wire from the outside. And so for any of you that might be familiar with electrical components or electricity, everybody that's in that field knows that an insulated wire performs much better than a wire that's just bare metal. And that's exactly what your neuron is doing. And so in these demyelinating forms, you're losing that essentially rubber coating around those axons, around those nerve cells that ultimately transmit the signal. In contrast to that, what we see in these axon degenerating forms of CMT is instead of just losing this myelin sheath or this rubber that wraps around the nerve cell, you end up actually degrading the wire itself. And so two different mechanisms, but ultimately the same outcome, a failure to communicate from your brain all the way down to your peripheral tissues, such as your muscle, and obviously resulting in the symptoms that we see across these varying forms of CMT. Now, in the context of CMT type 2A, as you'll notice on this slide, there are a whole lot of different mutations in a variety of genes that can lead to axon degeneration within CMT, and there's these axonal degenerating forms of CMT. However, in CMT type 2A, we're focused on one gene in particular, and that's a gene called MFN2 or mitofusin 2. And so mutations in mitofusin lead to this pathological change in what we call the mitochondria. And so these are fascinating organelles. I've built an entire career around mitochondrial biology and bioenergetics. And these are, for lack of a better nomenclature for them, we'll all probably remember them from high school biology as the powerhouse of the cell. And certainly they are exactly that. They produce energy for all of your cellular processes, including signaling within nerve cells. And we know that when you have these mitofusin 2 mutations, you end up having these changes in how these mitochondria interact with one another and how they ultimately form a network so that they themselves can function and produce energy. And ultimately what happens when you have these mitofusin 2 mutations is you lead to a failure of the dynamic regulation of these mitochondria within your nerve cells. And so you can think of this as the yin and yang, essentially, a balance that needs to be achieved. And when that balance is disrupted, you lead to things like impaired energy production and ultimately activation of pathways that lead to the degeneration of nerve cells. And one of those core components in those pathways is a protein that we call SARM1. And so SARM1 was really characterized about 5 to 10 years ago by doctors Jeff Milbrandt and Aaron D'Antonio at the Washington University in St. Louis. They identified this protein, and they found that it actually hydrolyzes this small little molecule that you have in your cell called NAD. And so you've probably heard, if you are anywhere on social media these days, you've probably heard something about NAD supplementation. And so NAD is a critical energetic substrate. And so when this gets depleted, you ultimately can have negative outcomes. And so that's exactly what happens when SARM1 is activated in response to mitochondrial injury, like you would see with mitofusin 2 mutations. You deplete NAD stores, and this ultimately leads to activation of other pathways that ultimately degrade these axons, these long projections from your nerve cells. Now, what's interesting about SARM1 is it's a protein that, for all intents and purposes, we've not really been able to identify a normal function. It's only bad whenever it is activated. Now, in most healthy individuals, this protein is in what we call a closed conformation. So it looks like a donut, essentially. And in response to these activating stimuli, like mitofusin 2 mutations in CMT2A patients, you end up opening SARM1 into what almost looks like a rose. It's a flower, if you will. And that allows the protein to have its activity, and you lose these substrates like NAD, as you can kind of see here. And so the thought process is, if this is the key regulator for this axon degeneration that we observe in diseases like CMT2A, and we don't really see a normal function for SARM1 in normal biology, why can't we just get rid of it, right? And so there were a lot of studies that looked at the genetic deletion of SARM1, and they found that in mouse models and rat models of disease, including CMT type 2A, that if you get rid of this protein called SARM1, you actually end up rescuing the CMT type 2A phenotype. And so the mice are actually completely cured when you knock out this SARM1 protein. And so the core thing that we were interested in doing is saying, well, in most humans, we can't really go in and we can't genetically modify this as easily as we could if we had just a drug that could go in and block its function. And so we looked at this in a very unique manner. There were other companies and other groups that were trying to target SARM1 by blocking that NADase function. So basically sticking a molecule right here to be able to block that conversion. And the problem with that approach was the fact that there's a lot of other proteins in your body that do this same phenomenon of hydrolyzing NAD. And so you could end up with off-target effects and potentially safety liabilities. And so we had the opportunity to design out a new molecule. And what we ended up doing is we said we can actually exploit the normal biology of this protein SARM1. We can take this protein, which undergoes this change, and instead of just saying we can put a molecule to block at this NAD conversion component, what if we just glued the thing back into an inactive state? And so that's what we eventually did through a lot of work, a lot of computational chemistry and biology. We were able to take activated SARM1. We built a molecule that acts essentially as Elmer's glue and is able to glue this protein back into an inactive state, thereby inhibiting its function. And when we actually tested this molecule, the first thing we did is we put it into a test tube. And in that test tube, we had this SARM1 protein, we had NAD, the substrate that it normally hydrolyzes, and we put drug in. And what we saw is, in a very nice dose-dependent manner, we were able to block the activity of SARM1. And so this was the first step at really trying to define out a drug program and saying, this is all fine and good, right? We can block this thing in a test tube, but does it have any actual effect on human cells? And so we took motor neurons that we had and we activated SARM1 chemically so that we could control it very nicely. And we used this compound called rotenone that induces the same mitochondrial damage that you would see with a mitofusin 2 mutation in a CMT type 2A patient. And what we saw is that when we treated with our drug, that we were able to maintain these axonal projections from those neurons. So we were actually indeed preventing those neurons from degenerating and ultimately leaving them in a functional state. Now, the next question, of course, is that's great, right? We saw a test tube, we saw some cells in a petri dish, but what happens when we actually look in a living organism? And so one of the most well-established models to date for this axon degeneration phenotype that's driven by SARM1 is in the context of amyotrophic lateral sclerosis, also more commonly referred to, of course, as ALS or Lou Gehrig's disease. And we know that they have very similar degenerative pathophysiology that you would see in a CMT patient with an axonal degenerating form of the disease. And so what we ended up doing is we treated these animals after they already had this peripheral motor dysfunction, and we treated them on Asha 624, our lead SARM1 inhibitor. And we saw that even after 6 weeks on therapy, we had a significant improvement in this animal's ability to move. So their motor function was being restored. And you can see that was significantly restored up towards what would be a healthy control animal by 12 weeks on drug. And you can measure this in a whole variety of different ways. And in all of those analyses, we saw significant improvements in the mouse's ability to actually move around and actually locomotion as a whole. And this was actually due not just to some symptomatic change that we were, seeing by improving, for example, neuron function by just allowing more ATP production, so energy production from those mitochondria that are being restored, like we showed earlier on one of the slides. But what this really translated to was a component that we call disease modification. And so this is where we actually looked at the neuromuscular junction. So this is the point in your muscles, in your legs, in this case, in the mouse, where the nerves actually meet the muscle and signal for that muscle to actually have a contracting phenotype. And what we ended up seeing was in ALS animals, there's a loss of innervation. So the green here is nerves as they approach the opposite side, the muscle side of where the nerve is innervating. The red, synaptophysin staining. This is actually staining the innervating nerve. And what we actually saw was, as known in ALS patients, and of course the mouse model, you lose innervation and you lose the architecture of this neuromuscular junction. It all just changes, it becomes bad, right? Disorganized. When we treated with Asha 624, we saw very clearly at the end of that 12-week treatment that we had a very nice organized neuromuscular junction, consistent with what you would see in a healthy mouse. We also saw a very nice reinnervation of that neuromuscular junction, and ultimately very nice overlap. Commensurate with that, we saw that all of those neuromuscular junction parameters were improved. And interestingly enough, a key biomarker for neurodegeneration or loss of neurons, a particular protein that leaks out into the blood called NFL, or neurofilament light, that is a key measure of degenerative phenotypes, was significantly reduced when we treated with Asha 624. So together, these studies really gave us promise that the therapeutic that we were developing, sure, was indeed inhibiting SARM1, but more importantly, that inhibition of SARM1 was having a very positive effect on both symptomatic improvements as well as disease-modifying improvements at the level of the biology that is actually driving the disease. And so we can see that we saw a nice reinnervation with 624 treatment in addition to that improved locomotion and obviously motor function as a whole. The other thing that was very, very beneficial in the context of Asha 624 that we were thrilled by was the fact that we were able to safely dose the compound. One of the biggest downfalls to most new drugs is that they end up failing when it comes to toxicology and safety. They may do exactly what you develop them to do from an efficacy viewpoint, but they ultimately carry risk factors that make them non-amiable to being an actual therapeutic medicine. So we tested this in a variety of settings. These are both acute toxicity studies where we give a very large dose of the drug once, all the way out to chronic toxicology where we would be administering the drug routinely for a long period of time, just like you would do in obviously a patient. And what we saw was that at therapeutic dose ranges, and actually well beyond therapeutic dose ranges, we've not observed any overt toxicity with the molecule. And again, we owe part of that to the fact that we specifically designed this molecule for SARM1, and it wasn't found by screening, hundreds of compounds or millions of compounds, I should say, to see what would stick essentially. And so we've selectively designed this molecule to be able to inhibit SARM1, and by virtue of that, we've been able to impart very robust safety parameters that we hope here in the near future will allow us to move quickly through safety trials as we start to move towards our human dosing studies. When it comes to CMT, we've been very blessed and honored to have funding by the CMT Research Foundation that was funded earlier this year. It's a multi-phase study that is focused on looking at the efficacy of Asha 624 in patient-derived cell models, as well as in animal models of CMT2A. So essentially replicating, hopefully, what we saw with the genetic knockouts of SARM1 in the animal models, and then eventually moving this forward to clinical utilization as quickly as possible is our main priority. And so, although the project was really just initiated, and obviously a lot of the reagents and resources that we're using in this project are currently being outgrown and differentiated on the cell side, and of course the the rat model is being bred to study numbers and aged, we have started to garner some preliminary data and gather this preliminary data, and we can see very clearly that in iPSC-derived motor neurons between healthy patients, those isolated from a CMT2A patient, and then, of course, those neurons treated with 624, we're seeing significant improvements in the overall viability of those cells. So how well do those cells actually survive when you plate them? And then ultimately, we're seeing improvements in restoration in the overall number of those axons, those neurites that are projecting out of those cell bodies. Those are the ones that are actually making those connections. And so we can delve even deeper into this in terms of the mitochondria within these neurons and within the axons and how they move and how much energy they're producing. And so those are all studies that we hope over the next few weeks we'll start to be gathering some very encouraging results on. But overall, we are extremely thrilled to being able to test Asha 624 in the CMT2A models. And we're very optimistic at this point based on what we've seen in other peripheral motor diseases such as ALS and drug-induced neuropathies that we will see an efficacious result both symptomatically and from a disease-modifying perspective. So on that note, I would like to wrap up there on this portion of the webinar. Laura, again, we are so appreciative of the opportunity that CMT Research Foundation has given us and entrusted us with. And I am more than happy to take any questions as we go through this evening. Thank you so much.
Great. Thank you, Bradlee, for that excellent overview of Asha's work targeting SARM1 and CMT2A. My name is Kate Anders, and I'm the Scientific Portfolio Manager for CMTRF's research team. For patients living with 2A, this treatment offers a key path forward in the way we can treat 2A and other axonal forms of CMT. SARM1 is well known in the neurodegeneration field for its role in axonal degeneration, which can lead to loss of the signaling output from our nervous system to our muscles. As Bradlee mentioned, SARM1 has been implicated in several other neurodegenerative conditions, including glaucoma, ALS, Alzheimer's, and CMT. By targeting this key pathway early, Asha's treatment can hopefully reduce any further axonal damage in patients with 2A. And by giving the nervous system support and time to recover from injury, this treatment can hopefully slow disease progression and rescue those neurons that are in the critical window between early stage injury and degeneration. This project could be a key breakthrough in the field's understanding of the molecular pathways that can be targeted in CMT2A and the best ways to tackle future 2A treatments for patients. At CMT2A, or at CMTRF, we're excited by the possibility of treatments for all CMT subtypes and how the work being done at Asha will propel the field in a new direction. We're now going to move into the Q&A portion of the webinar, so feel free to post any questions you have using the Q&A function. And to get started, one question that I had was, how often would patients need to take a treatment like this SARM1 inhibitor?
Yeah, no. So Kate, that's a great question. So what we're really anticipating based on the studies that we've done thus far in ALS, we're predicting on this being a single dose a day. So hopefully it will be obviously a pill formulation for most diseases in the context of other diseases like ALS. That sometimes has to be considered due to swallowing issues as an oral suspension. But this would likely be a once a day intervention.
Mm-hmm. And are there any effects of long-term SARM1 inhibition that you've seen in any preclinical assessments that you've done?
Yeah, so that's a phenomenal question. And that's always a concern on any type of drug program when you're obviously inhibiting something that is there. And the one thing that is unique about SARM1 is that we have not observed, both ourselves and all of the other groups that have studied SARM1 to date, that either complete genetic ablation or knockout of SARM1 or pharmacological inhibition of SARM1 directly has led to any off-target type toxicity or even on-target type toxicities that you would associate with blocking SARM1 function. There are some putative roles for it in the context of some very select immune reactions in the brain for some very rare neuroviruses that could occur. where SARM1 activation may be beneficial in actually restricting viral spread from one nerve to another nerve. However, that is an exceedingly rare complication that you would be exposed to one of those 4 very rare viruses. So we're very optimistic that long-term dosing and chronic inhibition of the protein is not going to result in any major detrimental side effect.
Great. So based on a couple of the questions that we have in the chat, so for those patients in the audience that are eager to be involved, can you comment on the timeline from getting to where you are in your current research stage to getting to treating patients or dosing patients, I should say?
Yeah, absolutely. So where we currently are is obviously we have multiple programs that we're looking at currently, for which will be the fastest way to clinic, right? We see with this particular molecule that, in addition to CMT2A and ALS, you have chemotherapy-induced peripheral neuropathies, you have diabetic peripheral neuropathy, all of these different, even more rare peripheral and optic atrophies, for example, and eye disease, all have SARM1 as a heavily indicated pathogenic mechanism. And so how we view this is ultimately it's almost a pipeline and a pill if it works, right? Where we have the ability to leverage it across multiple indications. And so from a clinical progression viewpoint, we're currently moving forward to IND enabling studies. And so what that really refers to is what we call GLP toxicology. So it's this next step of being able to take the molecule. We actually give that molecule to a third party, and they will test it in 2 different species of animal models for purely safety. This is not efficacy or anything at this point. This is not showing the drug is working against a disease. That's all been obviously done. This is now focused on the regulatory requirements for moving forward to a first-in-human dosing, whether that's through FDA approval, the European Medicines Association, or obviously in Australia is also a trial option. Our main focus is to move as quickly as possible so that we can start the safety component of the trials, which would obviously have to be completed first. Those are in healthy volunteers. And then from there, ideally within 8 to 12 months of finalizing that safety trial, you would be able to start dosing patients.
Thank you. Another question that based on what we have in the chat, so would taking this drug help or allow nerves to regenerate that have been damaged? And there's another question about, do you anticipate damaged nerves being able to repair with the dosage that you have here?
Yeah, so that's another phenomenal question. And that's $1 million question right there. So we see in some very select tissue culture studies, cultured neurons within a petri dish, inhibition of SARM1 obviously blocks further degeneration of those axons. But we have also seen that you do have regrowth of axons out into an open area, for example, on a tissue culture dish where we do a scratch analysis. And so there is thought process from a lot of scientists around, especially with peripheral nerves, so peripheral motor neurons at the level of the muscle, for example. We know those do regenerate. And from a biological viewpoint, inhibition of SARM1 that ultimately would support better bioenergetic function of those mitochondria that we talked about earlier will help to support an environment that is primed, if you will, for allowing regeneration to occur. We typically, out of an abundance of caution, don't go into saying that we are actually regrowing axons. However, there are studies that do suggest that if all of those components are in the right spot and you're in the right area, that's a possibility. So we're optimistic that we will see some sort of regeneration, even if it's very minimal. We hope that certainly is an outcome.
Great. So another question from the audience. So at this stage, is it looking like it would be useful for both younger and older CMT2A patients? Do you think that age could have any role in how someone would respond?
Yeah, so we don't have any indication that let's say a pediatric patient would respond any different than an adult patient when it comes to the drug itself and the potential efficacy of the drug from a viewpoint on the drug. What that really is meaning is if you're just giving the drug should do what the drug's supposed to do and inhibit SARM1. From a SARM1 inhibitory capacity, we do believe in that context that obviously the earlier that you would start therapy from a diagnosis, the more responsive that patient likely is going to be, right? That being said, a very late stage, advanced, older patient with CMT type 2A still could certainly benefit. This is something that we have discussions in the realm of ALS pathology as well, right? Are you dosing a patient that has been diagnosed from a naive state to a diagnosis within 4 or 5 weeks, all the way to a patient that's in, very progressive year 2 of the disease pathology? We see benefit in both scenarios. Obviously, the earlier that you can catch you can limit that much more degeneration, the better.
Yeah, absolutely. Another question we had was regarding target engagement biomarkers. So are there any biomarkers that you're utilizing alongside the studies that you're done that would be useful once you get into the clinical setting to see how effective your treatment is?
Yeah, so that's a very good question. So there's a variety of ways that we look at target engagement with SARM1. There is a direct biomarker for SARM1, and that's a protein called CADPR, or cyclic ribose. But CADPR itself can be measured. The problem with that is not going to be likely a broad-based biomarker for biofluids, so blood testing, for example, or CSF. Simply because those are very effective if you're looking at single neurons at a time. Beyond that, it's probably not going to be very good for a large-scale clinical trial unless Jeff and Aaron or other scientists develop new ways of analyzing it. That being said, obviously, plasma NFL levels, CSF NFL levels are a key biomarker for degeneration. And so that would be something that would automatically be included. And then more nuanced than that would be actually looking at markers of mitochondrial and bioenergetic function that we can look at. And so we do have an entire list of mitochondrial-based biomarkers and energetic biomarkers that do change when you obviously inhibit SARM1 and are restoring that upstream mitochondrial homeostasis.
Hey, Bradlee. We have a couple of families that are here because they have young children who have CMT2A. So they're asking about a tentative timeline. And in some of those cases, but also some other questions are about where will this opportunity be outside of the United States when the trial gets going?
Yeah, so that's a phenomenal question. So typically, again, how these trials typically would move forward is the first phase of this, regardless of location. Obviously, we would probably do it in the United States to stay in compliance with the FDA. We could also run it in Canada or Australia. Would be a phase 1 safety trial. And so this is a trial of healthy volunteers. They would have to be adult volunteers that will come in. They'll receive what we call a single ascending dose and then a multiple ascending dose of the drug. And so this is typically about a 12-week long trial that you're able to just make sure that the drug is safe and you're understanding the exposure levels of the actual compound so that you can effectively determine what dose you're going to give when you move to patients. Assuming we clear that, which we have no indication that we would run into any toxicity issues, so we're optimistic about that point. Assuming that gets cleared, upon initiating what would be a phase 2A type study in a larger patient cohort, there would be obviously some enrollment criteria along how we would do that. There's some thought process around if we move forward in CMT with a CMT2A only cohort, or if that gets broadened out to what would be essentially a basket type trial where you have multiple forms of axonal degeneration, subtypes being included. That's another option. And then, of course, if you do a parallel trial in something like ALS, can you include additional components in there? And so all of those questions are ones that we have pending with regulatory agencies, the FDA being one of those. And so when it comes to trial sites, that becomes a little bit more nuanced in terms of patient selection ability, how easy it'll be for those patients and their families to be able to get to that location. And so all of those things are currently under consideration. A lot of this will ultimately come down to what the FDA guides us on in terms of what they're going to be willing to approve or not approve. To the point of younger children, This is obviously where it gets even a little bit more nuanced. Typically, the FDA will not allow for a pediatric-based trial until we pass both an adult safety and then a pediatric repeat dosing safety study. Now, for something like CMT, obviously it makes sense that we would ideally enroll pediatric patients. Those are obviously the earlier stage diagnosed patients. And so that would be something that might push the timeline further because we would have to do a few components of safety in a pediatric population. But we've been in discussions with a variety of rare disease groups, including obviously CMT Research Foundation, as well as other pediatric rare disease groups on what those trials typically look like. They're not for SARM1, for example, in these other trials, but very well heeled with some key opinion leaders in the pediatric dosing space when it comes to pharmacology that are providing guidance. So we're optimistic that we can, with the right resources and backing, be able to move this forward as quickly as possible.
That's great news, Bradlee. And I'll let the families on this webinar know that as news unfolds on this, CMTRF will be updating you. So if you just keep in touch with us, we'll definitely be letting you know as all of this process works through. Bradlee, there's another good question around patients who are already using AFOs. Do you feel that this would be beneficial for them?
Yeah. So, look, I think there's definitely power in combination, right? And this goes across all neurodegenerative conditions, even your more mainstream Alzheimer's and Parkinson's. I don't see any reason that there would not be a potential for a combinatorial benefit, right? That being said, that is another caveat nuance with the FDA. If they will allow, in especially trial components, patients who are already on another therapy into the trial. And then, of course, once that drug is approved, then you do gain a lot of leeway with respect to prescribing in a combination with something like an AFO. So I think that's something that my short answer to that would be yes. I think it could be very beneficial. Longer answer, there's some nuanced complexity around it, but nothing that I don't think is non-achievable.
So I think another question in the chat. So you talked a little bit about this, about whether suppressing or removing the physiological function of SARM1 could induce adverse effects. I think there was another follow-up question saying that BEI at low concentration could paradoxically induce SARM1 activation. Do you have any comments on that?
Sorry, what was that in reference to? BEI?
I'm not 100% sure.
I'm okay. This might be a science. So I'm assuming they're meaning a base exchange inhibitor.
That's likely.
Okay, so I'm going to go science for a second. So apologies to the non-scientists. So yes, so there's evidence from Asha Therapeutics that was obviously acquired by Eli Lilly, their base exchange inhibitors. This was evidence published by Genentech from their program that these orthosteric inhibitors that bind to that NAD active site, when you have very low subtherapeutic levels of drug, you can actually paradoxically activate SARM1, which of course is not a good thing. So you're taking a scenario where you have active SARM1, you're inhibiting it, and then all of a sudden you paradoxically activate it. And so I don't think that's a program killer for those type of inhibitors. I think there's obviously ways to adjust dosing, and it's a lot more restrictive in terms of having to really define out that dosing. But that can be accomplished by maintaining, drug levels above a value that would induce that subtherapeutic dosing liability. That being said, we have tested Asha 624 using the same Genentech assay for that subtherapeutic dosing issue that has arisen with the base exchange inhibitors, and we do not see that subtherapeutic liability at all. So we've gone very, very low picomolar dosing, and in no instance have we actually paradoxically activated SARM1. That's a phenomenal question, though.
Another question from the chat. So would this SARM1 inhibitor also be effective on patients with ATP6 deviation, which I know is another mitochondrial disease?
Yeah, so I'm assuming this is an ATP6 mutation. We're doing some current studies. So I actually, interestingly enough, I help out as a philanthropic effort, serve as the chief scientific officer of Lucas Shy Foundation, which is focused on ATP6, AP2, and AP1 mutations and associated lysosomal and mitochondrial dysfunction. So we are actively testing it in that context to see if we can bolster actual mitochondrial function and even lysosomal functions as a consequence of that. We don't have any data yet on that. That's still all in progress. But nonetheless, there is potentially a benefit here beyond the axon degeneration, right? I think that's a key point. In ATP6, AP2, for example, or ATP6 mutations they come in a whole variety of flavors with a whole variety of phenotypic outcomes from hepatic insufficiencies and hepatic disease all the way through to Parkinson's-like movement disorder. And so there's certainly a potential for a therapeutic gain, and we're actively looking at it.
Another question from a participant: is there any point at which the disease may have progressed too far for the treatment to be effective?
Yeah, so that's a big question that we have really wrestled with, especially in the context of ALS pathology, which is of course exceedingly aggressive. We see it as if you can block further degeneration, right? Because becomes a cascade effect. You start with axon degeneration. That ultimately leads to more broadly injury and inflammation that ultimately now is, activating more broad neurodegeneration. And that progresses, right? It becomes a reactive cycle that repeats itself over and over. And so how we look at this is that even if you're in a later stage scenario, there could still be significant benefit from blocking further degeneration and taking those diseased neurons that are there and trying to bolster their function by restoring more upstream mitochondrial function and bioenergetics. So we would still see it as a putative therapeutic efficacy. Will it be curative? And we've talked about this to ALS patients as well. They're like, well, could I be able to stand up and walk someday? My honest opinion, probably not, right? I don't think we're going to regrow that many neurons when you're that advanced, but it could keep you alive, right? Stop your progression from that moment forward. And so that's really what we're hopeful for. If we see anything more than that, then that is obviously of just added value. But I think there is still value at any of the disease stages to intervene.
Great. Another question we had from a participant: so have you evaluated blood-brain barrier penetration, and is there like an extent where you think more would be beneficial?
Yeah, so the compound is blood-brain barrier penetrant. In the context of a lot of the CMT components, as well as obviously peripheral disease like ALS, if you're early enough, right, the peripheral exposures in the muscle, I think, are the key. At least that's what we see in a lot of the animal models that we have done to date. Now, certainly, as disease progresses and you start, really hitting both your lower and upper motor neuron components and into the brain itself, then yes, obviously, blood-brain barrier penetration and exposure is of critical importance. And so we do see that with Asha 624. We see very robust exposure. We do have an analog of the main drug that is actually peripherally restricted. And we wouldn't see any necessary need to use that unless for some reason there's a odd response occurring in the CNS. But again, we've not had any indication that would be the case.
All right, Bradlee, the questions are still coming, so we're going to keep going.
I'm good with it.
To what extent do you think this molecular glue inhibits SARM1 activity? Is it a complete 100%?
Yeah, so there's two ways to answer this. If we put it in a test tube, yeah, we can fully inhibit SARM1 activity. I think in the context of an actual in vivo setting inside of a body, right, whether that's a mouse body, a rat body, or a human body, you will eventually hit a component where you have exposure limits, right, that we're starting to hit a max tolerable dose from a safety viewpoint, and how much of that's actually getting into the tissue. So what we see currently when we look in the mouse models, we call it an EC95. So that's the effective concentration to see a 95% effect. And so we're able to reach that, right? And so whether that's a measure of, axon integrity, whether it's a measure of an actual behavioral readout like locomotion for like stride length, for example, we get to that point. So we wouldn't fully classify it as we have 100% of all SARM1 inhibited at all times. However, we do feel that we're hitting very good target exposure with the drug. And as a consequence of that, we're seeing, the efficacy that's associated with that.
That's awesome. Now, earlier, Bradlee, you answered a question about having to take this, like, is it one dose? Is it, and you said it will be a once a day. A question came in about, is that for the rest of a patient's life?
Yeah, so that is a good question. Our assumption is yes, it will be, likely having to be a durable treatment for the remainder of one's life. Unless, of course, something happens that fixes the initial insult, right? So the caveat there is for a CMT patient with a genetic driving component, chances are it's a lifelong therapy. For an ALS patient, you would see very similarly. If this is a patient that has had a traumatic brain injury, that might be a little bit different. If the drug's given early enough, and they're able to restore normal function as best as possible, you may at that point not have continual activation of SARM1. So an acute injury type scenario, possibly withdrawal. A more chronic setting would require likely durable dosing.
Thank you. All right. So this kind of came up a little bit, but it's good to refresh. Have you tested whether your compound might be effective in CMT2s beyond CMT2A?
We've not yet. So that is obviously part of our goal, is to expand beyond CMT type 2A. We're really focused currently on the funding that obviously we've been generously given by CMT Research Foundation to hit those key milestones on CMT type 2A first. And then we will expand out from there into additional axonal degenerating forms first. And then, our goal is to go even beyond that, right? We think there potentially could be an effect in a demyelinating form, right, where there might be mitochondrial deficit. And so we think it's worth a shot trying it there as well, even if it's not really biologically indicated.
All right, I think we'll go with one last question. So does your compound bind only to active SARM1 or to inactive SARM1 as well?
Yeah, good question. So as the molecular glue, the pocket that it actually is interacting with and then gluing the protein shut, it can interact in both active and inactive forms. So if the protein's already in the inactive state, it's basically binding and keeping it that way. If it's in the active state, it will bind and dissociate what we call the TIR domains. Those are the regions that actually form the rose petals, if you will, with that NADase active site that we were talking about. So it will inactivate both, keep inactive, and will inactivate the active form.
Awesome. Thank you so much. All right. So Bradlee, I can't thank you enough for taking the time to do this webinar to explain your work to this entire community, and then answering so many questions of just varied degree and background. It's just incredible. And thank you so much for choosing to work on CMT. The entire community is very grateful.